Hi there. Right, just move the little blocks around so I can start the slideshow. To wait a second. So we're going to talk about active transport today, which is a way of movement across the cell membrane. We were talking a little bit last session about diffusion and osmosis. So diffusion is considered a passive form of movement because it requires no energy, and so is osmosis. The movement of the molecules just happens naturally because the law of physics, essentially they're spreading out from where there's loads of them to where there's fewer. So it just happens naturally. It just spreads out as if the same way things spread out naturally. Like if you spill a drink, it will spread out naturally. Essentially, it's just like that. It's moving from a concentrated area to a less concentrated area. And what we're going to have a look at today is the opposite of that, when we need to move stuff from where there's not much to where there's already a high concentration of particles or molecules. Just going to have to move something on my screen because I can't get the slideshow button. There we go. So we're going to carry on with active transport. Before I start, let me double check you can hear me and I'm not talking to myself. Can you let me know if you can hear me okay? Perfect, thank you. Okay. So active transport then, it's movement of particles from an area where there are few to an area where there is already a concentrated number of particles. So it's movement of molecules or ions. I just tend to say particles, to be honest, because that covers everything. Movement of molecules or ions, you'll learn the proper definitions in them when you start chemistry. So don't worry if you're not sure what a molecule is or an ion or even an atom at the moment. You'll learn in chemistry. Active transport is the movement of these particles across the cell membrane. You can see the cell membrane on the diagram there in yellow. It's got that bilayer, that double layer, and they're moving things from a low concentration to a high concentration, to where there's not many stuff to where there's loads of stuff. So that's the way it doesn't want to go naturally. That's why we need to put energy in. So this process requires energy, usually in the form of something called ATP. And that's a chemical which gives energy in the cell. So sometimes people talk about it as the energy currency of the cell, but it's just basically a molecule that we can change slightly to release energy in our cells. So here you can see some proteins that go all the way across the membrane. And we talked about these a little bit when we were looking at the structure of the cell membrane a couple of weeks ago. So we can see these embedded proteins that cross the membrane there. And this is one of their roles as transport molecules. So this process requires ATP because the particles are moving across their concentration gradient. It's a bit like trying to move something uphill. You know, things will move naturally downhill because of laws of physics, gravity. And if you need to move something uphill, you've got to put some energy in because it's against the natural laws of physics. And that's what happens with the concentration gradient. If you've got a high concentration, they will naturally want to spread out. If you've got a low concentration, you're going to have to put some energy in to move them to somewhere where there's a high concentration. So how do we do this? Well, we use transport proteins. You can see these transport proteins in this membrane here. We often call them pumps because they work a bit like a pump. There's one called the sodium-potassium pump in animal cells, and there's a proton pump in bacterial cells and plant cells, but we won't worry about that too much. So we have this sodium and potassium pump that helps to move things across the membrane. So what this does is it helps maintain gradients that are needed to be kept a certain way. So a good example of this is in nerve impulses, and we'll have a look at this later on when we do the module on homeostasis, the unit on homeostasis. So nerve impulses, muscle contractions are dependent on ions being in a certain place. And it's also important for nutrient absorption because if you imagine we've got a load of glucose over here in the blood of the digestive system, and this was the inside of the cells. we'd want to get hold of every last bit of glucose possible. It's really important for our cells for energy. So we wouldn't just go, oh, right, you know, we've reached equilibrium. There's an equal amount of glucose in the blood to inside the cells of the gut, in the lumen. Let's just leave it at equilibrium. What we'd want to do is get every last bit of glucose possible. So we'd start to pump it across. So when we're pumping stuff across, or using energy at least to move stuff across, we can use different types of active transport. One type is called primary active transport that uses ATP, this energy molecule, directly. And then we have secondary active transport, which uses energy which is stored up in ion gradients that are created by this pump, essentially, where we use ATP. So we get a gradient of ions, so sodium and potassium ions, and we use that to drive other molecules or particles, substances, across the membranes. So we've got two types of transporter, if you like. We've got one that's called a symporter, where we use co-transport of two substances being moved in one direction, and we've got one called an antiporter, which you have two substances moving in opposite directions. We also have this uniporter, where something is moved in one direction. And something that we use, not active transport, but something else that we use these channels for, is for the movement of water molecules across the cell membranes. So this happens in animal intestines. Humans are obviously animals. When glucose is absorbed from the gut into the blood, so it can be transported around. There's glucose and sodium co-transport that's used for that. So those are your movements, your main ways of moving stuff across cell membranes, then: diffusion, osmosis, and active transport. Obviously, we had a little look as well at endo and exocytosis when we were looking at things being engulfed into cells, when we were looking at the cell membrane too. Okay, so last little bit that we need to do for this assessment criterion is disease and functional and structural disorders of the gut. So with the digestive system, we can have functional gastrointestinal disorders and structural disorders. So functional are disorders where there are persistent and reoccurring gastrointestinal symptoms. They occur as a result of abnormal functioning of the tract. So that's, you know, from what the medics say is from gum to bum. So it's from your mouth to the anus. That's the whole tract. You can get symptoms. So these occur as a result of abnormal functioning of the tract. They're not caused by any structural problems. So there's no tumors or masses or any biochemical abnormalities. So therefore, when you do medical tests and you're trying to examine why this patient is complaining of pain or discomfort, or something's not functioning properly, you can do CT scans, x-rays, etc., and the results will just come back as there's nothing wrong with you. It's absolutely fine. Obviously, there is something wrong because the patient is experiencing discomfort or pain, but quite often nothing will show up because there's nothing structurally wrong. Then, oops, just going to move a little box again so I can see the screen. There we go. Functional gastrointestinal disorders then can be a result of problems with motility. You'll remember from last session, we were talking about peristalsis, which is the muscular contractions of the circular and longitudinal muscles of the tube, so the esophagus and the intestines, but also we have the muscular activity in the stomach to churn everything up with that extra layer of muscle, that third layer there. So we've got the circular, longitudinal, and you've got the oblique going diagonally in the stomach as well. So these are muscular contractions, and you probably know if you've ever had cramp or anything in a skeletal muscle spasms and cramps can be really, really painful. So any abnormalities that's causing these muscles to not contract properly can be very painful, but also it can really slow down the movement of food through the gut, because that's peristaltic motion is what's pushing the food along. I think one of the main things that's important for considering with this is there enough fiber in the diet, because that's what the muscle pushes against, fiber, which is in the GI tract. So it's really important to ensure that the patient's diet is not causing problems. So another thing that is a feature of gastrointestinal disorders, functional gastrointestinal disorders, is sensitivity. So it's how the nerves are responding to the stimuli. So how they respond to digesting a meal, functional issues with the nerves. Sometimes they can be really sensitive. Neural pain is quite intense, and also obviously any sort of contractions of muscle can bring on pain then in the nerves if there's a nervous problem. There can always be also be brain gut dysfunction, which is when there's just a miscommunication really between the brain and the gastrointestinal system. So, you know, that could be for a number of reasons, like brain issues even. Irritable bowel syndrome is an example of this. So often patients have a lot of discomfort, pain, and it doesn't always show any results on tests, but it can be very difficult for the patient because the symptoms can be extremely painful, and they're also quite debilitating depending on how bad the symptoms become. Then the other type of disorder you get are structural disorders, and those in which the bowel does look abnormal and also isn't working properly. So abnormal functions of the bowel would tend to be things like diarrhea, constipation, as well as all of the pains and stuff we were just describing. So sometimes it's possible to remove structural abnormalities depending on what's wrong. We'll have a look at a couple in a minute, and you can see, well, that there's just lots of different types, and it's not always possible. It is possible to remove parts of sections of the bowel because it's so long. You can remove parts of the small intestine fairly easily, fairly routinely, parts of the colon as well. So it is possible to remove parts of it without too much difficulty. So common examples of structural disorders include hemorrhoids, diverticulitis or diverticular disease, colon polyps, colon cancer, and all sorts of inflammatory and bowel diseases. There's things like colitis and ulcerative colitis, which is an autoimmune disease which attacks the colon. There's Crohn's disease, which is an autoimmune disease where the immune system attacks the small intestine. So there's lots of different diseases which can cause problems in this area. And this is a vulnerable area because it is a exchange surface. It's a structure that has contact with the outside, like the lungs. They're really vulnerable to infection and inflammation as well. So here you can see diverticulitis in the image of the colon there, where you get these sort of enlarged lumps. And this particularly is really painful, that as well. So those are the differences between the functional and structural disorders of the gut. In the course content, there's more details on, because you've got to compare one functional and one structural disorder. So you choose which one you want to have a look in more detail at from the course content there, because there's quite a few. So that's movement of substances across the cell membrane. And obviously we've looked at the gut quite a bit there because that's one of the key places we want to move things across the cell membrane. Stop sharing this now. And if you've got any questions, you can pop them in the chat. Next lot of slides. Try that one. Hopefully it's that one. Where's it gone? Isn't it? Okie doke. Just get to the top of that. Right, okie doke. So what we're going to look at now, if there's no questions, let me just check. No, okay, is the liver. So the liver is what we call an accessory organ to the digestive system. So it's not part of the main tube, the gastrointestinal tract, but it does secrete stuff via the gallbladder into the digestive system, and it receives the blood, which is rich in nutrients as well. So it's got quite an important role. So glucose and amino acids and some fatty acids are moved from the ileum, which is part of the small intestine, into the bloodstream of the hepatic portal vein. We had a look at this the other week, and we said that these capillaries here drained into the veins. You can see the small intestine there, and drains into the hepatic portal vein, which takes the blood straight to the liver. So the liver is really important because it plays this detoxification role. So if you've eaten stuff like paracetamol or alcohol, it will help to remove these toxins from your blood. So what the liver is a really big organ, actually. It's tucked just underneath your ribs. And if you look at it at smaller level, it's made up in to these hexagonal subunits called lobules or lobules, depending on how you want to pronounce it. They are pretty complex structures. This is the diagram that you have. So you can see there, there's endothelial cells, which we've talked about in the first unit. There's hepatocytes, which are liver cells, so they're functional cells. We've got parts, we've got a tube there, duct for bile. So there's quite a lot going on. It's quite complex structure. So one of the really important functions of the liver is to produce bile to aid the digestion of fat. We talked about that last session. It increases the surface area of the fat and lipid droplets so that they can have a greater surface area for the lipase enzyme to work on. Another important role is it helps to synthesize and store fat, and it will also break down fats for energy. If we haven't eaten enough carbohydrates or don't have enough carbs, we can break down fats for energy. It also regulates cholesterol and lipoproteins in the blood. So quite important in terms of, well, in terms of what our blood looks like, if we've been eating lots of fat and things, and also to keep levels as they should be of certain types of lipids. So the liver, as we said last week, produces bile. It's stored in the gallbladder and released into the small intestine down the bile duct. The bile emulsifies fats, as I was just reminding you really, because we looked at it last week. So it breaks the large fat droplets into the smaller ones. This increases the surface area that the lipase enzyme can work on. So that's its like digestive role. This helps to digest the fats because it really massively increases the speed at which the enzyme can work, because the surface area of the fats or lipids or oils is increased. And it helps us to therefore absorb the fats out of our diet sufficiently. So it also synthesizes and stores fats. What it can do is convert excess carbohydrates and proteins. So if you eat a lot, into fatty acids and triglycerides. So it can store things, other substances that we eat. If we eat in excess, we can store fatty acids and triglycerides. We call this process lipogenesis. So lipo as in lipids, which is the name for fats and oils, and genesis is to make something. So if you kind of know these words, prefixes and suffixes and things like that, it makes it a little bit easier. So lipo means lipids, genesis means to make. So we're making fats essentially, and these fats can then be stored in the liver. We don't want to store too much fat in our liver, but we can store some without it being a problem, and we can transport it in the blood or to other tissues for energy storage elsewhere. They also break down fats for energy. So when the body needs energy, the liver can break down stored fats and release energy, forming something called acetyl-CoA. So don't really worry about the name of that for now, but it is an important molecule for energy releasing processes. And then another role is it regulates cholesterol and lipoproteins in the blood. So the liver produces, stores and breaks down cholesterol as needed. Now, we normally think of cholesterol as being bad because that's what we're sort of taught in terms of diet and things like that, but it's not all bad. And actually, cholesterol itself has got a very important role in stabilizing the cell membrane. So remember, we talked about the cell membrane. We looked at the video and saw it looking like a lava lamp. So you can imagine if that cell membrane was heated up a bit, that would become a lot more fluid and a lot more wobbly. So that's one of the roles of cholesterol. It stabilizes that cell membrane and stops it becoming too wobbly. But obviously it's quite a balancing act because we don't want too much because then it becomes too brittle as well. So the level of cholesterol is really important for cellular function. The liver also helps to maintain the proportion of HDL and LDL cholesterol in the blood, which is really important for heart health as well, because you'd like to have HDL, which stands for high density lipoprotein cholesterol, and we don't like to have too much LDL, which stands for low density lipoprotein cholesterol. So it's about keeping that balance, keeping that proportion right there. Okay, let's have a little look at some questions. How does the liver help regulate lipid levels? And which of the following processes describes the liver's conversion of excess carbohydrates and proteins into fatty acids and glycerol? Triglycerides, sorry. That was normally when you talk about fatty acids, you talk about fatty acids and glycerol. So I said it automatically. So have a little look at that, see if you can remember what we just said. And you can just write a note in your rough paper and I'll pop the answers up in a moment. If you want to, you can write on the chat on the screen as well. Okay, so what the liver does is it stores excess cholesterol and triglycerides, and it can convert excess proteins and carbs into fatty acids and triglycerides via the process called lipogenesis. So lipo or lipogenesis. Okay. Another thing that the glucose does is it helps to regulate blood sugar levels. So that's quite an important function. Obviously, if you're aware of anyone with diabetes, you'll be, does the liver really have 500 functions? Well, the liver enzymes have an awful lot of functions. So I guess if you started to discuss what the functions of the liver are in terms of the roles of their enzymes and the proteins and stuff, yes, there's a lot of proteins that are if you've ever been to the doctors and you get a blood test, you can pretty much almost always guarantee they'll do something called U&Es, which is your urea and electrolytes. That tells you how well your kidneys are doing. And they'll do LFTs as well, which is your liver function test. So it's a really key indicator of your health, how well your liver is functioning. It has got so many roles. I couldn't tell you specifically if there are 500, but it's a very important organ, and all of the proteins and enzymes that it produces are involved in loads of key functions as well. So yeah. So it also has a role in blood sugar levels, along with the pancreas. So what happens if your blood sugar is high, so you have higher than normal blood glucose levels, it triggers the pancreas, this specialized cells called beta cells in the pancreas, to release loads of insulin into the bloodstream. So this is the pancreas here, this sort of wiggly looking organ. And what this does is it stimulates the liver. So the insulin stimulates the liver and muscle cells to take up excess glucose from the bloodstream. You can see there's glucose molecules there. There are 6 carbon sugars, so we tend to draw them as hexagons. And then it is stored as glycogen. And the reason it's stored as glycogen is because storing glucose in the cell is quite dangerous. It would exert an osmotic pull. We talked about this a bit last week. It would result in excess water moving into the cells. We showed it with the red blood cells, but it can cause the cells to rupture because it will pull the water in. The glycogen, however, doesn't. It's much more, it's much safer for the cells. It won't pull the water in. So instead of storing glucose, we convert it into something called glycogen because it's osmotically inert. It doesn't pull the water. And this process, we're making glycogen. So it's called glycogenesis, genesis to make, glycogenesis, because we're making glycogen. There is only a certain amount of space we can store glycogen, and the rest of it's then converted into fat. So we need to do the opposite as well. If we haven't eaten for quite some time, we need to return that glycogen into glucose so that we can get blood glucose levels up around the cells, because we need a constant supply of glucose to keep us going. We need a constant supply of oxygen as well to keep us going. We need oxygen and glucose. Oxygen, fine, we can breathe. Glucose, not so easy because we don't eat constantly. So we store it and then release it out of storage. So if we get too much glucose in the blood, as we were just saying, the pancreas produces insulin, and that insulin, which I've drawn in green, will enter the blood. And then that allows the glucose to be absorbed by the body cells and the liver, so the muscle in the liver. And that means the circulating blood glucose is reduced. And then if we get too little glucose in the blood, which is needed for energy, cellular respiration, it's gone to too much. Is that the wrong way? No, I think I've got I think I've got the same bit then. But anyway, if you get too little in the blood, sorry, I haven't got the slides for that. They're the same ones that doubled up there. If you get too little glucose in the blood, like we've got here, the blood glucose levels falls, the hormone glucagon is released. And what happens then is that glycogen will be converted back into the glucose molecule, which can be used for energy. And then we call this glycogen lysis, because we are splitting the glycogen. So lysis means to split. So we have glucogenesis and glycogen lysis going on. Another thing that's important with this is adrenaline can cause more glucose to be released. So you can imagine you're in a fight or flight situation, you need to run, you're going to need more energy. Where are you going to get that glucose from? Well, you can get it out of storage. And that's one thing that adrenaline does. It will help you to release that glucose out of storage. The glycogen gets split back into glucose. In an emergency, if glucose is needed, fats and proteins can be converted into glucose as well, and that's called gluconeogenesis. So there's another process. There's loads of processes involved in keeping the blood glucose relatively stable so that we can keep moving, keep producing energy and stay alive while things are happening. So we've got lots of different substances with similar names. So we've got glucose, which is really important substrate in the cell. For respiration is the process when we're going to talk about that later. It helps us to stay alive, helps our cells do things. We've got glycogen, which is how we, animals, humans, store glucose. And we've got glucagon, and they all seem quite similar words. And glucagon is the hormone that will convert the glycogen back to glucose when we need it in an emergency. That doesn't happen that much these days because we've got good access to food, to be honest. It's very rarely that you're not going to have glucose circulating. You'd have had to have not eaten for quite a long time. Okay. So let's have a look at these, see if you can remember what was the role of the liver in blood glucose regulation. When blood glucose are high, what does the liver do? And what do we call the process of converting glucose into glycogen? So have a look through and see if you can remember. This is quite a lot. This is a tricky topic, I think, because the words are all so similar. So I think it makes it quite confusing. But just remember that genesis is to make, lysis is to split, and that helps quite a lot. Okay, well done. I'll give you a minute and then I'll pop the answers up. So what does the liver do with blood glucose regulation? It stores and releases glucose as is needed. So stores glucose after a meal when you've got loads in your blood, releases it when you haven't eaten for a while and there's not so much in your blood. It's the pancreas that's involved in producing insulin. If blood glucose levels are too high, the liver will regulate this by converting glucose into glycogen, which is how humans, animals, store their carbohydrates. And the process of converting glucose into glycogen is called glycogenesis. So genesis, to make, glyco for glycogen. Okay, I'll skip through those because I want to make sure we get through a little bit more. So we're going to have a look now at how hepatocytes or liver cells clear the blood of some toxins. And one of the things that the liver does is, and this is an important process, that it's called deamination. So last week we had a look at the structure of proteins and we said they have a chain of amino acids which can get twisted into a helix shape, folded into a zigzag shape, and then that chain will fold up and you can get multiple chains stuck together. And that will give you a protein, a globular protein. And we know that we eat proteins, it's part of our diet, and that blood from the small intestine, this is the lumen of the small intestine here, and the blood that gets supplied with the nutrients, the blood goes into the liver from the small intestine. So when we eat proteins, what gets absorbed is not the protein. You might remember from last week that it gets broken down by the enzymes in the stomach, and we absorb the amino acids. So the amino acids are absorbed into the blood, and they go via the hepatic portal vein into the liver, and then they have to get broken down. So this is the chemical structure of an amino acid, and they all have this group here, NH2. So you can see an N and two H's, NH2. So that's called the amine group, and we need to remove it. So that process is called deamination, and that takes place in the liver. And it actually creates something toxic to the body called ammonia. And that very quickly has to get converted into urea, which is much safer. And then we pass this urea to the kidneys and it's excreted as urine. So really important role is deamination of the amino acid, and that's removal of that NH2. And obviously, then we've got this urea to get rid of via the kidneys. So let's have a look at this amino acid. I've just flipped it upside down there. And you can see on this molecule here, we've got a carboxyl group on this end. So this is where this OOH is. You can see it's there, COOH. And then we've got the amine group here, NH2. NH2. You can see on this is a process that happens, deamination, urea is produced and off it goes to the kidney. So there are 20 amino acids in human nutrition, or possibly 21. And if we have a look at them here, you can see this NH2 group, NH2, 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 NH2. Don't worry too much about the structure of these. But what's important is you can see that all of these amino acids have got this NH2. Around half of these can be synthesized in the liver by a process called transamination. And that basically offers an alternative route for the amine group rather than ammonia, urea, and excretion. So we can take this amine group, this NH2, and we just saw on the previous slide that we can convert it to urea and it goes off into the urine via the kidneys, or it can be moved onto another amino acid and build a different amino acid out of the bits that you've got from the old amino acid, essentially. So this diagram, you can see that it adds the amino acid to the top right-hand corner. And it's basically rebuilding amino acids. So quick look at the role of the bile then. I know we talked about it last week, but since we're on the liver, let's go over it again. It's produced in the liver, stored in the gallbladder, which is adjacent, and then it will secrete, or it will be secreted from the bile duct into the small intestine. And we did mention last week that one of the things that the bile does is neutralizes the acidic stomach contents as they enter that small intestine. So it's not just the emulsification, but it's the neutralization of this really strong acid is another function. Don't forget that we want to create the optimal optimal pH for the enzymes of the small intestine. And this is one way we can do it. One way we can bring that pH up from the really acidic conditions from the stomach. So the other thing that it does is it emulsifies the fats and oils, as we spoke about last week. So we're splitting up these larger fat droplets into much smaller fat droplets. So the volume is going to stay the same, but the surface area is going to increase. So that means it's much easier for the lipase enzyme to work on all of the oil and fat that we've consumed. So volume stays the same, surface area increases, much larger surface area available for that lipase. And that helps to break down then the fat that's in our diet. So this is considered to be an exocrine function, which is to release something out into the blood or directly rather than down down a duct of the bile duct there. So liver, number one, bile, gallbladder is this little ball. And then here we've got the duodenum, number two, and the duct is going straight in there at number two into the intestine. So bile is produced in the liver, stored in the gallbladder, released down into that duodenum for those two functions of breaking down the fat, emulsification, and also neutralizing that content. So let's have a look at this. Which is the process by which alcohol is broken down? What is the substance broken down during deamination? What effect does insulin have? And what effect does glucagon have? Yeah, good work. Well done. Um, Ethanol is an alcohol. Ethanol definitely isn't good for our bodies. But alcohol's actually not good for our bodies. I know it's like culturally quite significant in most cultures, including most European cultures, but it is a neurotoxin and it is actually toxic to pretty much every cell in your body. So yeah, it doesn't do us any good, but it is a toxin essentially. Okay, let's have a look then at the answers. So we break down alcohol by detoxification and other substances as well, any other drugs that we shouldn't have, well, I shouldn't say shouldn't have consumed, but have consumed, like paracetamol, need to be broken down. Amino acids from our protein in our diet are broken down during deamination, and we can convert them into urea, which is then excreted in the urine, or we can recycle them and remove that amine group onto other amino acids. The insulin is released from the pancreas, but it can decrease blood glucose levels by causing glucose to be stored in the liver, converted and stored as glycogen. And glucagon can increase blood glucose levels by directly stopping the take-up. Okay. Right. Any questions on that before I pop the next slide up? Actually, I suppose what I should ask as well is, did you find the content on movement across the cell membrane okay after last week? Does anyone need to go over that again? Because if there's a lot of you, I could go back over that content rather than rushing on to the next bit if you'd prefer. Sure, I'll pop the next one up then. Okay, give me a sec. Right. Okay. Let's have a look at the next bit. So outline the protection of immune factors and removal of pathogens from the blood. So the liver has also got an immune role, and it is one of the sites of antigen presentation, which we will talk about later on when we get onto the immune system. So that's a really key role for the immune system. It has these special cells. You can see them here, Kupffer cells. And obviously the blood flowing via the hepatic artery is going to pass through by these cells. Remember, these were the structures that we see in the liver. And there are immune phagocytotic cells here. And what these do is they will engulf and remove any unwanted particles in the blood. So think about where this is situated. The liver is the first place the blood is going to go to after small intestine. So small intestine, you could be eating all sorts of horrible pathogens, which could be from people's unwashed hands when they've been preparing your food. They could be from your unwashed hands while you've been preparing the food. They could be from contamination of the food itself. You might not have washed the food. Farms tend to use animal feces to fertilize their fields. So there could be all sorts of contaminants on your food. So your liver's got a very, very important role there of checking for stuff that you've eaten and has managed to get across your gut. So what happens with these cells is they phagocytize and engulf foreign particles. So it could be bacteria it could be pollen or dust more in the respiratory system. It's unlikely, a bit more unlikely in your digestive system. But bacteria and viruses, stuff that you can pick up on your hands. We call it the fecal oral route, which is obviously not very pleasant because it's the transfer of fecal particles that contain fecal bacteria into your digestive system. But that's a very common route of entry for pathogens. So this immune system role of the liver is really important because of its place as the first place that the blood goes to once it's got to the small intestine. So we have had a look at the role of hepatocytes in carbohydrate, fat, and protein metabolism. And we're going to have a look at how the immune system factors in the removal now. So it's obviously, we know that the liver is important in metabolic and detoxification processes. So the metabolic was the removal of those amine groups to produce the urea and the transamination. And the detoxification is how the liver will remove things like alcohol, but it's also got this really important immunological role. So here you can see what we were talking about last session again. And I think this is probably about the third time I've showed you this slide. And this is the key reason I keep showing you this slide is the situation, the position of the liver, the first port of call for the blood after getting all of those things from the environment. So the food, the drugs, the alcohol, the bacteria, anything that ends up in our system, the first place it's going to go from that exchange surface of the small intestine is the liver. So this is, again, why it's so important. So first place that the blood goes from the gut, and much of this immune activity occurs in these specialized capillaries of the liver, and they mix with the blood from the hepatic portal vein with the hepatic artery. What's going to happen is the blood is going to flow past these specialized cells that you can see over here. So these Kupffer cells, and we've got something called the hepatic arteriole here. And this is the sort of bit larger than a capillary. So it's a slightly bigger blood vessel than we've got over here. And we've got these specialized macrophages right here mixing with the blood. You can see the structure of the macrophages where they've got that sort of pseudopods, we called them last week. You can see them clearer here, but they can go out and engulf these pathogens. So these cells are long-lived, so they hang around for a long time, which is relatively unusual for the blood cells. We're usually replacing them at quite a high rate. So they're adhering to the endothelial walls. So they're actually attached, which is also unusual for blood and immune cells. They tend to be circulating, and this allows them to filter the blood as it actually goes through. So they're kept in situ, in place, in the liver. They're attached rather than just sort of traveling around as the blood moves around the body. So they engulf and destroy any bacteria, any parasites, and any other microorganisms that we're getting straight from the hepatic portal vein. They will also take away any aged or damaged red blood cells and cellular debris. So this is quite an important function of the liver as well, to get rid of the red blood cells. Red blood cells don't live very long or circulate for very long. They've got a very short life. So they need to, there's a really high turnover of these cells. So they need to be removed really regularly. Once these things have been phagocytosed, brought inside of the cell, they're then broken down with special enzymes which are found within the cell. So they'll be brought in and digested and broken down within the cell. What these specialized cells do, these Kupffer cells, is they secrete cytokines. These are things that will activate other immune cells. They promote inflammation. When an infection is detected, they cause inflammation, which is like an activate signal to the immune system. And they also help to initiate tissue repair responses for damaged. So that is quite an important immunological role. Cytokines are something that really sort of trigger like your immune response and how bad it is. Because you might have heard, I don't know if you remember that well, or with that key engaged when COVID was going on, there was a lot of talk about cytokine storms and how some people were really badly affected by the illness compared to other people. And they started to hypothesize that it was cytokine storms were being triggered in some people, which will cause a really big immune response. So how these cells respond during an infection is really important. The liver, as we were saying earlier, is constantly exposed to the harmful molecules and microbes in the gut because of its position, because it's next to the exchange surface of the gut. So it helps to maintain something called immune tolerance, which stops these unnecessarily large immune reactions against stuff like dietary antigens, stuff that you might be eating that obviously you could react to, and other bacteria. Commensal bacteria is bacteria that's meant to be there, it lives there. So it's important that these Kupffer cells and their cytokine secretion is kept at a sort of regular level that doesn't go over the top and start off a really big immune reaction that causes a lot of damage. So summarizing then what's going on, we've got the Kupffer cells, they filter the blood, they're fixed in position, they remove pathogens, debris, old blood.
Catch that. Could you try again?
I'm sorry, Siri again. So they remove the old red blood cells as well. That's really important. That actually gets the debris from that gets some of it ends up in the bile as well. They produce cytokines which will determine to a certain extent how the immune system is triggered. and they help to maintain immune tolerance as well, so we don't overreact and start having allergic reactions almost from other things in the gut. So they're both defenders and regulators, and that regulator role is very, very important. The immune system is very susceptible to going wrong, as we'll see later on when we talk about autoimmune disease. It can very seriously overreact as well during infection, as we've seen in various epidemics. So, for example, certain people, the way they responded to COVID, Spanish flu, is another example where the immune system caused overreaction or extreme reaction, caused a lot of fatalities in people. So this immune role is not just important as acting as a defender, but it's important that it keeps everything calm and not too large of an immune response as well. So the next thing we need to have a look at is how the hepatocytes is liver cells, clear the blood of drugs and other toxins. So as we are well aware, it is a detoxification organ, and it plays an important role, particularly in removing alcohol and drugs such as paracetamol and metabolic wastes that we were talking about earlier with the protein metabolism. So all of these nutrients, again, they go in through the digestive tract. First place they go to, hepatic portal vein, is the liver. So again, hence the reason for its importance. So the hepatocytes have an awful lot of the smooth endoplasmic reticulum, and this is really important for detoxification reactions. So you might remember back when we were looking at the structure of the cell, the smooth endoplasmic reticulum was this big membrane that's close to the nucleus, but it looks smooth because it's got no ribosomes attached. The hepatocytes, so the liver cells, also have an awful lot of mitochondria, and they are really important because mitochondria supplies ATP. It's the energy for the cell. Remember, we were talking a moment ago about ATP being used for active transport. So that is why these mitochondria are really important. Produce the ATP, all of these metabolic processes that need stuff to be moved across the cell, or energy for anything else. It's all available on site. And then they have this huge network of enzymes. So they are involved in so many processes, oxidation and reduction and conjugation reactions. So they're breaking stuff up, they're sticking stuff together. Really important and a key indicator of health, actually. Like I said earlier, one of the first things that gets tested when you go to the doctors, if you need a screen of bloods, is your liver function. I think we should probably think about winding it up then rather than getting into this, because we're in the next part of we won't get through that by the end of the session. So let's go on to that next session. Keep it on that slide. Right, has anybody got any questions about the liver before the end of the session? Yeah, it is a tricky topic because there's so much stuff going on. And I know that the amino acid stuff can look a bit scary because of the biochemistry, but yeah, definitely go over it. It's something there's a lot of information, isn't there? In this, there's a lot of content to get through. So definitely have a read through it. Okay, guys, if you haven't got any questions, have a good evening, and I will see you on Thursday.